2008 group problems. Question 1 said, suppose an individual is heterozygous for the following inversion: normal chromosomes, all capital L, M, N, centromere OPQRSTUVWX, and the inversion is lowercase L, M, N, the centromere OPQVUTSRWX. And it says, what are the four products of meiosis if there's crossover between S and T? Well, you'll have the two normal ones that are there, and then after the crossover, you'd have an LMN centromere OPQRSTUVQPO centromere NML. So that one's dicentric, and then you'd also have an XWVUTSRWX. For question 2 is fill in the blanks. Non-disjunction in meiosis 2 produces 50% aneuploid gametes, whereas non-disjunction in mitosis produces the mosaic phenotype. I said for part B, meiosis in individuals who have suffered a paracentric inversion produces a dicentric chromosome and an acentric chromosome. For C, Turner syndrome is an example of a human disease caused by monosomy, while familial Down syndrome is an example of a disease caused by Robertsonian translocation. Human liver cells can be 4N, 18N, or 16N. A terminal deletion on chromosome 5 causes the Cry-Ducat syndrome, while an inversion on the X chromosome can cause the Marie tooth decay. For part F, gene duplication is a change in a chromosome structure that can be potentially beneficial to an organism or an entire species. And then for part G, when a translocation cross is formed, the alternate segregation pattern can produce viable gametes and offspring. For C, it asks how many chromosomes would be in humans with the following conditions. For Turner syndrome, it would be 45, Jacobs 47, the Cry-Ducat is still 46, familial Down syndrome is 45, and then type A hemophilia is 46 still. For part B, it says which one's not associated with the Charcot-Marie tooth disease. It is the ones that not associated are triploid, aneuploid, reciprocal translocation, trisomy, and non-disjunction. The one that was associated with it was diploid and duplication. For part C, which of the following terms are associated with Klinefelter syndrome? That's when you have two X's and a Y. So that would be aneuploid, trisomy, and non-disjunction. Part D, it said which of the following terms are associated with type A hemophilia? I said none of them are, because it's normal, which would be diploid, which is not an option, and it's also duplication, which wasn't an option. Or an inversion, sorry, which wasn't an option. For part 4, so it says we have a DEFGH centromere IJKL chromosome with MNOP centromere QRST, and it says suppose reciprocal translocation occurs between one chromosome and each pair, producing DEFP chromosome QRST and also MNOGH asterisk IJKL. So it says diagram these chromosomes in prophase 1. So I just kind of wrote them right next to each other for that first part. For part B, it says identify the allele composition produced by alternate segregation. So you'd have the two normal ones paired together, the DEFGH IJKL and the MNOP QRST. And then you'd have the two switched parts paired together, the MNOGH IJKL DEF QRST together. For part C, identify the allele composition D for adjacent 1 segregation. So I got the DEFGH IJKL with the DEFP QRST and the MNOGH IJKL with the MNOP QRST. Then for the last part, it's identify the allele composition of the gametes produced by adjacent 2 segregation, which is DEFGH IJKL with the MNOGH GHIJKL, and the MNOPQRST with the DEFPQRST. And then the last part says, cats have 38 chromosomes. How many chromosomes would be found in the following cats? A triploid cat have 57. A monosomic cat for chromosome 12 is 37. They just lose one. For part C, if it suffered a terminal deletion on chromosome 7, it's still 38. Part D, if it has a Robertsonian translocation on chromosomes 17 and 18, they're combined, so it's at 37. And then a reciprocal translocation between 6 and 9 still leaves it with 38 chromosomes. And that is the genetics group problems lesson 8.